Okay, so this is chapter 15. So we're going to be taking a look at senses. And so when you take a look at your senses, the first thing you need to understand is that we have to basically wire your senses so that you can receive information. We do that with what's called sensory receptors. So basically, these will convert whatever the information is coming from the environment, essentially a stimulus, and it will basically relay that into a nerve impulse, which will then move through transduction. So this is the conversion component to it, to the central nervous system, right? So you basically go from this kind of conversion strategy, and then that gets relayed to your central nervous system, your brain, so forth. So you have extra receptors are essentially responsible for detecting outer stimuli. For instance, your main senses, your taste, smell, vision, hearing, equilibrium, those are all extra receptors sensing your external environment. And they're constantly sending information to your central nervous system, and you're sorting through all that, making sense of it all. Right. So essentially, your different receptors that you're going to see here are your taste cells that you're going to see, your taste buds, your olfactory cells in your olfactory bulb, the rods and the cones in your retina for seeing, the hair cells in the spiral organ of your inner ear. Those basically are for sound waves. That's hearing in the ear. Your semicircular canals, we'll take a look at all these guys here in this chapter, which is essentially your equilibrium, your sense of equilibrium. That's typically rotational. So how do you know when you're rotating? That's your semicircular canals. And then an open region called the vestibule is your gravitational equilibrium. And these are all different types of receptors. Some of them are what's called chemoreceptors. In your eyes, it's photoreceptors. And in your ears, it's mechanoreceptors. And so we'll take a look at the different types of receptors here in just a little bit. Interoceptors, so these are basically looking at internal stimuli. So a good example of this would be things like baroreceptors, whose job it is to sense blood pressure. Also osmoreceptors that measure or sense the differences in the your chemoreceptors, like you had with your taste and sense. These guys in particular in the interoceptor group are specifically focused on pH in the blood itself. And so these guys are the ones who are maintaining homeostasis. So the way that you maintain homeostasis is that you monitor all these different parameters, and then you make your adjustments to maintain homeostasis. So if you get a little too high in your blood pressure, you make some adjustments to bring it back down. If you get a little too salty in your osmoregulation, then you kind of add a little bit of water to bring it down, or you get rid of the salt. So you can kind of adjust it with your homeostatic feedback mechanisms, especially your negative feedback mechanisms. So we can classify these guys in a couple of different ways by what they do, right? So first of all, we have the chemoreceptors, photoreceptors, mechanoreceptors, and thermoreceptors. So the chemoreceptors are basically responsible for sensing chemical substances. For instance, your sense of taste, smell, your blood pH, those are all types of chemoreceptors. A subdivision of the chemoreceptors, and a special case of chemoreceptors, is what's called a nociceptor. So the nociceptor is essentially your pain receptor. So it's a type of chemoreceptor. And that's because typically what you're going to see is you'll see dendrites, naked dendrites. That's basically your neuron dendrites. If you recall, the dendrites are the little hair-like projections basically sticking out from the cell body. So those are the dendrites. So these naked dendrites will basically respond and attach to different types of chemicals that are usually released by damaged tissues. And then this will create a sensory response that we'll then associate with pain. That tells us that there's something wrong in the body. It alerts us to danger. It alerts us to a problem that's happening in our body somewhere. Then we have our photoreceptors, which typically will be photosensitive, so they'll respond to light. They'll be activated when they're exposed to light. Mechanoreceptors will be activated when they are mechanically distorted. For instance, your sense of hearing and balance is all mechanical distortion. So is touch. Blood pressure can be. Stretch receptors, those are a special type of receptors that are sensitive to mechanical stresses. And what's called proprioceptors for your proprioception. Those are all types of mechanical receptors. Basically, they respond to physical distortion of some sort of a structure. Okay, so bear with me. I know I always have to do this every single thing, but I've had enough ups and downs with these systems to warrant that I need to do this. So, good. Everything looks good. So we'll just keep on going. Thermoreceptors, the last one. These guys are going to be sensing changes in temperature, especially important in regulating your body temperature. For instance, in your hypothalamus and your skin, you've got thermoreceptors that are constantly gauging your body temperature, and then they're able to make an appropriate response after that. So when you take a look at sensation, right? So sensation is not just collecting data. Sensation is also conscious perception of that data, the stimulus, so to speak. This is happening in the cerebral cortex, right? So ultimately, what's going to happen is the sensation will be sorted out with the appropriate area of the brain that receives the nerve signals, right? These are these integration or association areas. So essentially, a sensory receptor will basically take in the nerve signal, and then they'll carry out the integration. And then what'll happen is your brain will make an association with that in the association domain. So you basically get a stimulus that's coming in, usually through the peripheral nervous system. Your sensory receptor will pick up that stimulus. It will relay that into the nerve. And so the sensory nerves will bring it up to the brain or to the spinal cord if it's a reflex. The spinal cord will then relay that to the brain if it is not. And then the central nervous system will make some sort of a decision, and then basically will send that back down in terms of its association or the sensation if there is no appropriate response for it. It's just, I feel this and I just want to make some sort of an association with it. So one type of integration that your brain can do is what's called sensory adaptation. So this is basically a decrease in your response over a repetitive stimulus, right? So to a stimulus over time. So for instance, if you smell some sort of an odor at first, then oftentimes you may not even be aware of it. It's like your brain is just basically getting used to it and subtracting out that data because it's already made an identification there. So the sensory receptors then will basically send fewer impulses to the brain because it realizes that the brain is essentially ignoring it because it's something that it already knows or understands, and it wants to move on to something else that it doesn't know or understand. So now we have somatic senses. Basically, the somatic is associated with the muscles and the joints. The skin is also part of that, and of course the viscera. These are the organs in your body. So this is your somatic senses. And so we have three different types of somatic senses. You have what's called proprioceptors, cutaneous receptors, cutaneous meaning your skin cover, and of course your pain receptors. So all of them basically will send their nerve impulses through the spinal cord to a area of the cerebral cortex that is the primary sensory area of the cortex itself. So the area where basically you receive that incoming information and you essentially do all the integration and understanding of what type of a response to send out for that. Proprioceptors. These guys are typically mechanoreceptors, and they're often mostly involved in reflex reactions, especially in muscle tone, equilibrium, and posture. So these are kind of like background activities. So for instance, a specific type of proprioceptor called the muscle spindles are embedded in muscle fibers. And so what happens then is if the muscles will relax too much, that basically means they stretch out. This will send a nerve impulse through these muscle spindles to contract the muscle so you don't overstretch. So that's a type of proprioception. The Golgi tendon organs, this is basically going to be in the tendons. So essentially, this is where muscles get stretched too much. Then they're going to generate and fire that nerve impulse, and this will cause the muscles to basically respond and to kind of get themselves back, essentially. So if they stretch out too much, you'll send a message to them saying, okay, listen, you can't just totally fall apart here. You got to keep yourself a little more tightly wired. We have to basically be functional. So when you take a look at the muscle spindles and the Golgi tendon organs, these will maintain muscle tone. Basically, it basically maintains a state of readiness. And this allows for more efficient contraction. So a good example is your knee jerk reflex. This will involve your muscle spindles, and it maintains your equilibrium, your posture. So this is what it looks like. So in the first step, basically, what's going to happen is your muscle spindle fibers will basically contract and kick, causing that kind of inner response of that sensory inputs. So it's going to take in that sensory information, going through that dorsal root ganglion. You know what's going to happen because it's a reflex. It's going to go through the interneuron and it's going to go out the ventral side to the muscle fibers, and that's going to basically cause you to kick. And so that's basically what it looks like. So the Golgi tendon organ you can see is this similar sort of a sensory situation, this proprioceptor that you see kind of at the beginning of the tendon, right when the muscle turns into the tendon itself. So let's take a look at cutaneous receptors. So cutaneous receptors, basically, this is something that will allow you to be able to sense touch. For instance, you'll be able to sense pressure, pain, temperature, and also deep and light touch. So the types of cutaneous that we have is what's called Meissner corpuscles and what's called Krauss end bulbs. So this is in your fingertips, your palms, your lips, your tongues, your nipples, the penis, and the clitoris. So this is basically creates very fine touch. Fine touch sensation. So it's very gentle touch. The Merkel discs, which is another thing, this is also in your skin. This is basically kind of at the interface where your epidermis meets the dermis. And so these are also touch receptors, a little bit deeper, not quite light touch. And the root hair plexus, basically, there's a free nerve ending that wraps around the hair follicle. This basically responds to when the hair is touched. So you can actually feel when your hair is touched without actually touching your skin. That's because you're activating that root hair plexus. And so this is kind of what it looks like. So you can see structurally, first of all, the Merkel discs, which are touched, they're basically kind of right here at the interface of the epidermis and the dermis. The free nerve endings, like little nerve twigs, which you basically have up here in the epidermis itself, will also be there for things like not necessarily touch, but for pain and heat and cold. So you can feel that and sense that quickly and right away when it touches you. The Krauss end bulbs, which are also touch, are a little deeper into the dermis. So you have to have a little bit of a deeper touch for that one. The corpuscles that are right underneath the epidermis, these guys are going to be your Meissner corpuscles. They're usually light touch. And then you have deeper, larger corpuscles called the Pacinian corpuscles, which are deep pressure. And then every now and then you'll see this little group, these kind of what's called Ruffini endings, which are also pressure sensors. So you have a couple of series of pressure sensors, a lot of light touch, deep pressure, more crude touch. So you have to basically have a more crude touch to get to the Krauss end bulbs, for instance. And then you also have hair touch, which is basically these nerves that are wrapped around the hair shaft, the root hair plexus. Okay, so the cutaneous receptors will have two types, right? So first of all, one will have sensitivity to pressure. For instance, this is going to be your Pacinian corpuscles, which is kind of an onion-shaped looking structure in the dermis, deep, typically in the dermis, and it's for deep pressure. And then the Ruffini endings are encapsulated by connective tissue. This is for lighter pressure because they're higher in the dermis. So you can sense different levels of pressure. So those are the cutaneous receptors. The thermoreceptors are basically free nerve endings in the epidermis, right? So these typically are going to be cold sensors and hot sensors or heat sensors to allow you to determine whether or not you have a cold or a hot object touching your skin. The pain receptors are a little bit different, right? So oftentimes these will be turned on when you have tissues that are damaged. They'll typically release chemicals that will stimulate these pain receptors, or toxins, which will be released by bacteria, which will stimulate these pain receptors. You can also have mechanical distortion, which can trigger them, thermal distortion if it's hot, too hot. Electrical stimulus can also activate these. And what they do is they release a chemical called prostaglandin, which is a type hormone, and these guys will go and bind to the nociceptors. And so oftentimes when you take a look at a pain medication, like for instance, aspirin or ibuprofen, these will reduce pain because what they do is they inhibit the enzymes that actually go and make the prostaglandins. So you're essentially taking prostaglandins out. So you're eliminating the communication of these prostaglandins with the nociceptors, which essentially is what causes or creates the pain. So referred pain is essentially something where we have the stimulation of the nociceptors, right? This is what causes the pain. Oftentimes in internal organs, but it'll oftentimes be felt as pain in the skin also. So it's basically not the location of the actual stimulus. We've all experienced this, right? So for instance, if we have pain from the heart, is oftentimes felt in the shoulder or the arm. And so sometimes if we say, for instance, scratch one part of our body, it feels like we feel a little nervous tinge in our shoulder or something like that. So that's referred pain. And so the nociceptors will also receive impulses from the skin, which is the reason why those two are linked together. So you get pain in one, it can also reference that to the skin. Which basically is an illustration of how integrated our nervous system is. So then we have taste and smell. So we're going to be taking a specific look at taste and smell, right? So these are the chemical senses because their receptors are sensitive to molecules, for instance, in the food or in the air. The chemical receptors themselves are basically in the plasma membrane of the cells, and they bind specifically as receptors to specific molecules. For instance, a chemoreceptor can be divided into two different types of receptors, those that will basically touch or receive distant stimuli, for instance, your olfactory cells, right, basically born on the air, versus those that are direct stimulus, which are essentially direct contact. Things like your taste cells and your pH receptors. So those are basically registering different types of sensations that are directly contacting them. So when you take a look at your sense of taste, we have it organized in structures called taste buds. So taste buds basically are located on what's called the papilla, which are basically bumps that you see on your tongue. So if you take a look at your tongue, you'll see these kind of like bump-like structures on your tongue, and your taste buds are on those bumps. You also have a few of these on your hard palate and the pharynx and the epiglottis, but for the most part, it's on your papilla of your tongue. And so we have 5 main taste areas or receptors for taste, right? So the so-called taste zones of the tongue, we've all probably seen this in varying degrees. We can taste sweet, sour, salty, bitter, and a new one that was most recently discovered, umami, which is a savory, kind of a meaty type of a taste. So we taste meat with the umami reflex. So this is actually an electron micrograph of the actual dermal papilla, of the papilla of the taste bud. So you'll actually see these large structures here are the actual papilla, these fleshy projections of the tongue. and then you'll see a little taste bud that is basically projecting off of the papilla. So that's the taste bud. And it's on the taste buds that you have the receptors. Okay, so when we blow it up, so here's a taste bud coming off of the papilla. So you can first of all see that before you even get to the structure, you notice you have a sensory nerve that basically comes in here and will actually innervate each of the pieces of the taste bud, right? Similar to the way we saw it innervating muscle fibers, it's going to be innervating the taste bud itself. So the taste bud will have a couple of different cells. First of all, it'll have what's called a supporting cell, so which is a cell that kind of supports the actual taste bud itself. This is the one that's connected to the nervous connection. And then, of course, the taste cell, which is the one that's going to be responsible for the actual taste piece. And then at the surface, you'll see what's called a taste pore. So your molecule will come in here, and then it'll basically land, go through the taste pore, it'll land on the actual taste cell, and then that will trigger a nervous response that will then be associated by your brain. And so typically you'll see these areas of the tongue, so the taste of your tongue, these buds, these papilla with these taste buds spread out all over your tongue so you can taste what you're eating. So this is actually a good image because it kind of pulls together your entire oral cavity. So you can see here, you've got your tonsils in the back, and then your epiglottis. So it kind of sees, you can see a nice structure of the tongue here. So the taste buds will open at this taste pore, which will basically allow the molecules to come in. And so they're supported by supporting cells and taste cells. So they're a combination of supporting cells and taste cells. They have what's called microvilli, which are small, tiny hair-like progressions, hair-like structures on the surface. So you can see these like little hair-like structures out here on the surface of those support cells. And so those are important because when you take a look at those guys, what's going to happen is the molecules of your food are going to come in and bind to the receptor proteins on the microvilli. It's going to cause a nerve signal to be sent to the what's called the gustatory or taste cortex in the parietal lobe. This is where you're processing that information. And so they're then interpreted as a particular taste. So your brain, remember, is the one that makes the interpretation of the taste. Your tongue is just simply collecting data, right? What you do with that data or how you associate with that data is happening in your association area of your brain. So let's take a look at sense of smell. So basically, if you think about it, our sense of taste and smell kind of go together. Matter of fact, the overwhelming majority of your sense of taste, quote unquote, is actually your sense of smell. And so we can illustrate this like if we plug our nose, we can't really taste food very well. If we have a really bad cold, we can't really taste food very well. We can't really taste it until we actually smell it. So what happens is we have in our nasal cavity olfactory cells found in the, what's called the olfactory epithelium. That's at the very roof of our nasal cavity. This is basically where you have modified neurons from the olfactory bulb, which is in your brain. And these olfactory cilia, which have these receptor proteins for odor molecules, are hanging down from the roof of your nasal cavity. And so this is your top of the roof of your nasal cavity, and up here is where your brain is. There's your crista galli of your ethmoid, and then you have your brain up here. So your olfactory bulb is a nerve that basically lands right on top. It's a cranial nerve that lands right there on top. It's a little nervelets basically dangle down through these little holes in that skull bone right there. That's where the receptors where the odor molecules are. And then those have the olfactory cells. So this is kind of what it looks like. So here is your olfactory tract. This is your olfactory nerve. This is in the brain. So up here is the brain. This is the skull bone. This is what's called your ethmoid bone in your skull. It forms the nasal cavity. So you can see how you have different tracks within your olfactory bulb that basically move through these little holes in the ethmoid bone. And then what they'll do is they'll come in and they'll basically kind of dangle at the roof with these little microvilli sticking out, these projections, these olfactory cilia sticking out there with these receptors in them, waiting for you to smell an odor. So you have these like little hair-like extensions in the roof of your nasal cavity. It's all embedded and taken care of in a cell layer. So they're protected by this cell layer that you have here. That's your olfactory epithelium. So what will happen then is this will be in contact with the air, and these molecules will stick to the receptors, and then that will trigger the nerve response, which will then come back. The reason why we lose our sense of smell when we get sick is because what happens is this epithelium in your nasal cavity that produces mucus will produce lots and lots of mucus, which will start to cover up these cilia, and it basically disallows the air molecules to actually make contact. So you kind of can't smell anything until you clear out all that congestion and it re-exposes these olfactory cilia so you can actually smell molecules again. So when you smell odors, basically this is going past an olfactory cell. And you have about 1,000 different types of receptor proteins on your olfactory cell. And so ultimately, these nerve fibers will basically go back to the same nerve, the olfactory bulb, that's the cranial nerve. And so an odor that contains a lot of different molecules will basically kind of turn on or activate a different combination of receptor proteins. So it's kind of like playing a piano, right? A different combination of keys will create a different sound. And so a different spectrum of activations of these receptor proteins will create a separate type of a sensation or a smell sensation. The olfactory cortex then is the association region for your olfaction, located in the temporal lobe. And so the olfactory bulbs will basically connect to the temporal lobe for association. That's what your brain does with that. So this determines whether or not you like that smell or you don't like that smell. So this is an association of your brain, and it's different for each person. So it also connects directly with the limbic system, or that's the seat of your emotions. So that oftentimes you can link emotion and memory to a particular smell. Smell can be very memory stimulating. Now, as we get older, typically the number of olfactory cells begins to decline with age, which means our sense of smell begins to wane a little bit. Okay, let's go ahead and take a look at the eye. So the eye basically is our dominant sense, composed of three main layers called the sclera. We also have the choroid region, and then we have the retina. So the retina, I'll just number it now. So the sclera is basically the outer layer. It's a tough, fibrous connective tissue layer. It's the white of our eye. The only place that's not white is the cornea, which is clear so we can see through it. Now the choroid is the middle layer. It typically tends to have a dark pigment. It absorbs light that the photoreceptors have not absorbed. This basically reduces glare and reflection of light that you're not focusing on. It extends on the front side to become the iris, which is in the job of the iris, which is the colored part of our eye. The job there is to regulate the size of the pupil, which is simply an opening. This is where light enters into the eye itself. And so it thickens a little bit just beyond the colored portion to create what's called the ciliary body. And this will basically control the shape of the lens for focusing. So the iris controls the size of the pupil. the ciliary body, which is the thicker base of the iris structure, is for focusing the lens. This is kind of what it looks like. So here you can see, first of all, the sclera, which is the white part. Number 2 is the choroid. And then number 3 is the retina. And so if you take a look at the sclera, you can see the sclera goes all the way around. That's the white of our eyes. And then when it gets to the cornea, it becomes translucent. And the cornea is kind of like a translucent, clear bubble, if you will, over our eye to protect the eye. And then just below the cornea is going to be a fluid-filled cavity. Called the aqueous humor. If you take a look at the choroid, you'll notice that as it basically comes around to the edge, you'll notice it kind of starts to thicken here. And then the thicker piece of it here, this thicker club part of it, this is the ciliary body. Notice how it's attached to these ligaments. So these are the suspensory ligaments that are attached to the lens. So this is your lens. And so as the ciliary body contracts, it'll basically pull the shape of the lens to basically focus the light. The front end of the ciliary body will basically extend beyond and cover those suspensory ligaments. This will be your iris. This is the iris part, or this is the colored part. So behind the iris is your suspensory ligaments. So they're there to protect you. And then in the middle of the iris is your pupil. It's basically just a hole allowing light to pass through the lens. Okay. And one other piece that we forgot to mention, a couple of things, is behind the lens is this large open chamber. This is what's called the vitreous humor. It's full of a jelly-like fluid that helps to focus the light and refract the light. On the back of the retina is a little spot called the fovea centralis. This is basically where you focus your image. And then at where the inlet of the optic nerve comes in is basically where your blind spot is, because you have no rods or cones there. Okay. So the lens, which is attached to the ciliary body by the suspensory ligaments, basically divides the eye into 2 basic compartments. The anterior compartment, which is in front of the lens, that's where the aqueous humor is, and the posterior compartment behind it, which is where the vitreous humor is. So the anterior compartment is full of the aqueous humor, and the vitreous humor is basically part of the posterior compartment. But basically, the aqueous humor is constantly going to be produced and drained by very, very tiny ducts that lead into this particular area. What this does is basically create lubrication for the inside of the cornea, so that the cornea doesn't have any kind of chafing effect on the underlying iris or pupil or anything of that nature. This also basically creates a lot of support and cushion, so shock absorption, if you will. Right, in glaucoma, basically these ducts that are draining aqueous humor tends to get blocked, and it builds up in this aqueous humor. This creates a lot of pressure. And so when you build up the pressure in this chamber, it doesn't really have anywhere to go except for these drainage ducts. And so the pressure just starts to build up and build up and build up. And then what happens is it starts to push on the arteries that are in there serving the retina. This is also where the photoreceptors are located. And then slowly what will happen is you'll start to starve photoreceptors. They'll start to die. And as they die, basically you'll get loss of vision, and eventually they'll go to total blindness. So the retina then is the third layer. It's the posterior compartment. It's going to be the big chamber is going to be filled with vitreous humor, and the liner on the back of the wall of the inner eye is going to be where the retina is. So the vitreous humor is this jelly-like fluid that will hold that retina in place. It's not attached like feathers, and so it's basically held in place by the vitreous humor. And in the retina are going to be your photoreceptors, the so-called rods and the cones. Now, the rods are important because what the rods do is they basically are sensitive to light levels. So what I think of is radiant rods. So it's black-white contrast. That's what you see with your rods. Your cones, I always think of colorful cones. This is your sense of color perception. So basically you need bright light to see this, and this will allow you to be able to see colors. And so simply, if you don't see color, it's because your cones are not there. Like a person who's red-green deficient with color blindness, it means they don't have those cones to be able to see that. On the back of the retina is a structure called the fovea centralis, which we pointed out earlier. This basically is an area that's rich in cones, so lots of color perception. And so ultimately, when we look at an object, it's basically focused onto the fovea. So the fovea is like our screen. This is where we cast the image. That we see. Now, right next to that is the optic nerve, which is basically full of sensory fibers that are coming from the retina. This is going to take the nerve signals to the visual cortex in the occipital lobe for processing. So this is basically a table of the different structures that we just talked about. So I don't want to go over this. This is just for your personal study. So when you take a look at the lens, the focus of the lens is to basically focus the image, right? So the cornea, the lens, and the humor, the aqueous humor, all work together to focus the image on the back of the retina. So when the light rays are coming through, they're bent or refracted, and then they're brought into focus. So that's kind of what focusing does. So the image on the retina is actually inverted. So it's an upside down image. And so it's going to be reversed from left to right. So your brain actually has to turn it upside down in order to turn it up. So when you basically take a look at the lens activity, you have what's called visual accommodation. This is your adaptation to close vision when you're seeing things up close. So what's going to happen is the lens is going to change its shape to focus it onto the retina. And that shape is going to be driven by the ciliary muscle, which is in that ciliary body that we just looked at. If you a distant object, the ciliary muscle is going to be relaxed, and then the lens flattens out. That allows you to be able to see distant. To see a near object, the ciliary muscle has to contract, and that basically tightens the lens up, and it becomes round and thick, so you can actually focus on the close objects. Now, since close work requires the contraction of ciliary muscle, it can oftentimes start to fatigue. This is known as eye strain. This is one of the reasons why if you're reading a lot, you're doing a lot of close eye work, you want to look up a little bit, and you just kind of want to break out of that eye strain piece. You probably felt it, like if you've been reading a lot and then you look up and it's like everything's kind of blurry and you're kind of like your eyes are trying to readjust. That's eye strain. So you get a lot of this after the age of 40 becomes more pronounced. Talking about eye strain, I'm going to put my glasses on so I can help myself out here. And so this is oftentimes also because the lenses tend to lose elasticity, and so it's unable to sort of shorten and fatten and contract the way it was when we were younger. So when you do eye drops, for instance, basically this helps to reduce eye strain by lubricating the system, or if you use corrective lenses, then you can also reduce eye strain to help basically not make your eye do so much of the work. You're allowing your glasses to do some of the work as well. Here you can see that what's going to happen is you're going to focus your light rays are going to come into the eye, and they're going to basically focus, go through the lens, they're going to bend, and they're going to basically focus the object on the back of the retina, right on the fovea. Right, that's that fovea centralis. That's your screen where you're projecting it. And then when you have closer objects, then you'll notice you basically get the same thing, only in this case, you get kind of a slightly different focus. It's a bit of a larger focus. So this is on a near object as opposed to a distant object. So something that's far away, will come in and you'll basically focus it on the back of your retina. Notice how the lens is thinner here and narrower, and then for an object that's near, it's basically fatter and thicker, and then you're still going to be able to actually focus that image on the back of your retina, right on top of it. So rods and cones basically have a couple of different segments. They have an outer segment and an inner segment that's joined by a short stalk. Right, so they're inside them. There are pigment molecules, and so these are going to be in the membrane of the outer segment. So we'll take a look at this here in just a second. We also have synaptic vesicles, where we talked a little bit about that before, that are located in the synaptic endings of the inner segment. So we take a look at these guys. So this is basically a type of photoreceptor. And so you'll see this far one here is the rod, and the close one here is the cone. But you'll see what's referred to as a bipolar neuron. So here you have your synaptic endings, your cell body, and then this is your inner segment, and then the outer segment, which typically has ion channels of the membrane that are basically going to be receiving the stimulus. So that's what the rods are going to be doing. These guys receive the stimulus. The same thing is true for the cones, where you have the inner portion of the neuron itself, bipolar, and then you have this kind of receptor membrane disc that's going to be receiving that incoming stimulus. And so here you can see you've got your rods here. This is an actual scanning electron micrograph of the rods, and in this case, you have the cones. So now let's go ahead and take a look at our visual pigment. All right, so in our photoreceptors, we have a visual pigment in the rods called rhodopsin. Now, when a rod absorbs light, rhodopsin, which is a pigment, will split into two different structures, one called opsin and the other one called retinal. It's a derivative of vitamin A. This is the reason why vitamin A helps you to see. So once you split this, then there's a cascade that's triggered. And ultimately, you'll release an inhibitory neurotransmitter from the rod. So basically, the rod is inhibited, and then this inhibitor is released. So you kind of remove the inhibitor that's normally inhibiting the rod, and that ceases. So the rod's able to be active. Now, the rods are sensitive to light, and therefore these are your night vision images. So the more rod you have, the better night vision you have. The fewer rods you have, the less night vision you have. So we have rods everywhere in the retina except for the fovea, which is cone rich. And so this will give us a lot of our peripheral vision. A lot of peripheral vision is just crude movement and also contrast vision. So this is kind of like the two different types of photoreceptors that you can see here. So here is your membrane disc that we saw in our rod. So the light rays will basically come in and they will split the rhodopsin molecule into retinal and opsin, right? So that's what's happening here. So you're getting the split of these two molecules in here. That's going to create this cascade of reactions that leads to the ion channels to close, and thereby allowing the stimulation of the rods. The cones, which are mostly in the fovea centralis, that's where the bulk of your color vision is, that's where your cones are typically activated or turned on by bright light, and they can detect very fine grades of color. So for instance, we have three different types of cones. We've got green, blue, and red, our primary colors. And anybody who's into art knows that with those three colors, you can make any color you want. And so when we combine these cones, basically we can see different colors and different shades of colors and different hues. Now in some animals, you don't have all three colors. Maybe you'll have one or two colors. So the the retina itself has 3 different layers associated with it. That's just the retina. So the choroid, the one that lays next to the choroid, is the one that contains the rods and the cones. So that's the closest one to the choroid. Then you're going to have a layer of bipolar cells, which covers the rods and the cones. So those are those neurons that we talked about before. And then the innermost one are what's called ganglionic cells or ganglion cells, right? And so these basically will feed information from the rods and cones into the optic nerve. And so only the rods and cones are the photoreceptive ones. So those are the only ones that are photosensitive, not the ganglion. The ganglion are waiting for the information from the rods and cones to relay that to the optic nerve. Remember, a ganglion is essentially a processing center that's composed of lots of cell bodies. So your original sensation comes from the rods and cones. It's relayed to a processing center in the ganglions, in the ganglionic cells, and then that information is then sent on to the optic nerve. And so this is kind of what it looks like. So here you can kind of see your layers. So here's your sclera up here on top. That's the white of your eye. The choroid, which is the darker region that leads ultimately to the ciliary muscle and the ciliary body and the iris. And then underneath that, you've got your retina. The first layer is going to be this rod cone layer where you see all the rods and the cones basically associated with your sensation, right? And then underneath that, you're going to get these bipolar cells, which are these guys right here. So these are those bipolar neurons. And then below that are going to be your ganglionic cells. So that's these guys right here. So these are ganglionic cell layer. So those will do the processing of the information, and then they'll send that out from the ganglion on to the actual, on its way to the brain or to the optic nerve. So you do a little bit of processing of the information before you send it on. Now your blind spot is basically where your optic nerve meets the eye. So at the blind spot, because it's all just nerve, you're just conducting stimulus to the actual optic nerve. There's no rods or cones there, so there's no sight. You can't see anything. That's your blind spot, right? So when you have your two eyes together, you basically complete your vision, right? Because the blind spot of the right eye is not the same as the left. So your right eye covers for the left and your left eye covers for the right. So you don't have a blind spot. If you want to test your blind spot, one of the things you can do is take your fingers from the side in your peripheral vision and slowly start to move them in. And then at one point, you'll start to see that your finger disappears. That's your blind spot, and then it'll reappear. And then basically you just move through your blind spot. So that's basically your blind spot for your left and your right eye. That's how you test for it. So it's kind of what it looks like. So here you can see in this area right here where the optic nerve is coming in, there's no cells here. So you notice here is your 3 layers of retina with your bipolar, your ganglion, and your rods and cones, but that doesn't happen here. So here, basically, you're just relaying all that information and it's going into the optic nerve on its way to the brain, to your optic association area in the brain. Now, once the nerve impulses are moved into the optic nerve, it's going to enter what's called the optic chiasma, which basically means cross. So basically what happens is you have your 2 eyes like this. You have your optic nerves coming out of your eyes. What happens is your optic nerves come out of your eyes, and then in your brain, they basically cross like that. That's your optic chiasma. So it basically kind of makes a cross like that, and it goes to opposing areas. So this basically will cross over and then go to opposite sides of the brain. And so after you exit the chiasma, typically they become the optic tracts, which is essentially what we refer to. A tract is like a nerve. Only in the brain. So we usually refer to nerve outside of the brain and its tracts when it's inside the brain. So the optic tracts will then go to the thalamus, and then eventually those signals will go to the visual cortex and the occipital lobe. Here's what it looks like. So here's your information coming from one eye to the next. So you can see basically you're going to get this crossover of the optic chiasma. So it's going to cross over to the optic chiasma, and then it's going to relay through the thalamus and then go to the occipital lobe, and the other eye will do the same thing and go to the occipital lobe. So you're basically processing this with the opposite eye. So your left eye is going to the right side, and your right eye is going to the left side because it crosses over in the optic chiasma. So what does two-eye vision give us? Well, what it gives us is good depth of perception. For instance, if you want to challenge yourself with this, just cover one eye and try to gauge the distance between you and some other object. It's a lot harder. And so this gives you good depth perception, something that a lot of animals don't have. And so what happens is when we have this good depth, we also have to make some judgments there. But the image on our retina, remember, is inverted and reversed. So when it goes to our brain, we have to basically correct it, right? So we have to basically right the ship. We have to turn it up, right side up. Then we have to turn it side to side. So we have to basically invert it. And this will allow us to be able to correctly perceive it and to interpret it. So what are some abnormalities of the eye itself? Well, first of all, we talk about blindness. This is a genetic disorder. Oftentimes it's a red-green deficiency, so you have the inability to see red or green. And so this is generally an X-linked disorder. It basically means males will get it from their mothers, and so it will only be in males. And so, well, usually it's in males. Technically, females can have it, but getting a female is rare. So normally we're talking about maybe less than 10% of the male population will have a red-green deficiency. And it's always going to be a gene for one of the cones, basically, on your X chromosome that just happens to be deficient, and you're not able to discern red and green. What about distance vision? So basically, if the way we define 20/20 vision and what 20/20 means is if you take a standard of 20 feet, so if you can see from 20 feet what a person with normal vision can see from 20 feet, you are said to have 20/20 vision. That means my 20 is a normal 20. And a person who's nearsighted, they can see close, but they can't see far, right? So oftentimes what happens here is the eye becomes elongated. And so the distant objects are brought into focus in front of the retina. So the image actually falls just short of the retina, creating blurriness. When we have to see distant objects, we have to wear a concave lens. So concave, remember, is the bulge outward, like that. Convex. So this would be concave side. The bulge inward would be convex. So you have to have a lens that kind of bulges outward a little bit. This basically will spread those light rays so that the image will land on the retina. So you're manipulating the light rays. So this is kind of what it looks like. So for nearsightedness, I'm taking in an image, and then it falls just short of the actual screen, if you will, my retina. And so it basically is out of focus. Now, if I wear a lens like this, then what's going to happen is it will basically readjust that, refocus, and kind of rebend the light so that it casts the light. That's what corrective lenses will do. Okay. So my eyes were nearsighted since when I was like in college. Unfortunately now, as I get older, so farsighted is basically when you can see far, but you can't see close. So I'm losing slowly my near as well. So, which is typically something that oftentimes happens. So it's not uncommon for that to happen as you get older. It's what most people end up doing. In this case, your eye is shortened, and so the close objects are focused behind the retina. So it's like you're casting the image a little too far. So in this case, you're going to be wearing convex lenses, which will basically increase the bending of the light rays so that it readjusts that image onto your retina. So it's kind of what it looks like. So normally you can see how in a far-sighted situation, you're going to be focusing that image and it's going to be right behind your retina. So it's a little out of focus. And then the convex lenses will basically bring these right back into line with your retina so that you can actually see. And that's what lenses are doing. It's basically it's bending the light so that it can get that image to cast on your retina. So to be clear, now here's a question. Why isn't it casting on the retina? Why is in nearsightedness, is it falling short? And in farsightedness, is it going too long? It happens to be because of the nature of the manipulation of your lenses. So the musculature of your ciliary muscle over time begins to get weaker and weaker, and it begins harder and harder to focus that light appropriately on your retina. And so as it becomes weaker with age, oftentimes, then you can focus your lenses, but it doesn't focus perfectly. And so it's your ciliary muscle that is beginning to sort of have a hard time focusing correctly. That's what's happening there. Astigmatism is basically where your cornea or maybe your lens is a little on the uneven side, creates kind of a little bit of a fuzzy image. And oftentimes this can be corrected with uneven lenses. So you can get lenses with an astigma correction to them. So you can see here in this particular situation where you have your light coming in and the nature of your focal point is basically kind of a little skewed. So it's basically trying to focus this light. It's first of all going to be blurry because it's not getting to the retina, but the other one is it's off target. So it should put it on the fovea. It's not, it's kind of more over by your blind area. That's because of the unevenness or a little bit of a swelling either on your lens or on your cornea. This happens with scar tissue and things like that. In this case, what they can do is they can basically kind of create a lens with an astigmatism correction. So they can put that and have that light go through the astigmatism. So it's not basically messing up the bending of the light. And so that'll get it back on the retina appropriately. Okay. So now let's go ahead and take a look at hearing. And then we'll finish up with hearing and balance. So the ear has two functions, hearing and balance. So hearing and equilibrium, and the sensory receptors are basically in the inner ear. So both of them are going to be in the inner ear. The important cells that you see here are what's called hair cells, which have stereocilia, stereo because of the sound, cilia because they're little hairs. And so these are long, kind of stiff microvilli, so they're sensitive to mechanical stimulation. They're designed to distort. And the stereocilia are a type of mechanical receptor. So they're basically designed to distort, and they basically create the type of sensation that you need. Now, in the ear, there's three different divisions. There's the outer ear, the middle ear, and the inner ear. The outer ear is associated with the pinna. That's the piece that you can grab onto on the outside of your head. The auditory canal, which is where you put your finger into. So this is going to be lined with hairs and modified sweat glands that secrete earwax. That's ceramin, right? That's a type of wax called ceramin. And so this basically is designed to protect you from foreign matter coming into your ear. The middle ear begins at the tympanic membrane, which is your eardrum, okay? And this ends at a bony wall containing two small openings covered by membranes, one called the oval window, the other one called the round window. So between the two is going to be what's called the auditory vesicles. These are three small bones that are in the middle ear. The malleus is the hammer, which is the one that's attached to the tympanic membrane. The incus is the anvil, which is attached to the malleus. And the stapes, or the stirrup, is touched or attached to the oval window on the other side. So these three basically move together as a unit. So here's kind of what it looks like. So here is your outer ear. This is the piece that we largely encounter out here. So here is your auditory canal. And so you're going to basically move the tympanic membrane, which is going to move the malleus. And then you have the incus and then the stirrups for the stapes. And so as your tympanic membrane moves, it's going to move these auditory ossicles, which is going to move this oval window membrane that the stapes is attached to. And then you have your inner ear. And in your inner ear, you have your cochlea, which is this large snail-like structure. And you have your semicircular canals up here, all connected to your cochlear nerve and your vestibular nerve. So the auditory tube, or the eustachian or pharyngeal tympanic tube, is what connects the middle ear to the nasal pharynx. So if you take a look at this guy right here, the auditory tube is connected to the nasal pharynx. This is oftentimes the reason why really bad congestion in your nasal cavity can plug up your ears, because it's plugging up this eustachian tube. And so the other thing is also, since it's air filled, you can oftentimes equalize air pressure across this membrane. So for instance, when you go up to the mountains, pressures will change and they'll build up in your eustachian tube. And then what you do is you release the pressure of your tympanic membrane to equilibrate to your outside environment. So where the outer ear and the middle ear contain air, the inner ear, however, is filled with fluid. And so there are three areas of the inner ear: the semicircular canals, which are important, the vestibule, which is the base of the semicircular canals, and the cochlea, which is associated with hearing. So then ultimately, then through the auditory canal and the middle ear, sound waves will strike the tympanic membrane, because all sound waves is air waves that are compressed, that are hitting your tympanic membrane. This will cause your membrane to vibrate back and forth, which then pulls on their malleus, incus, and your stapes back and forth. And then what they will do is they will pull on the oval window. So these auditory ossicles, the malleus, incus, and the stapes, will amplify the vibrations. And then the stapes will strike or pull on the oval window, kind of like a wave machine. And then what it'll do is it'll cause the oval window to vibrate. And then this will create pressure on the fluid that's just behind the oval window. And this will recreate waves in the fluid, in the inner ear fluid. And leads to the cochlea. So the cochlea has three major canals. One that is called the organ of Corti, or the spiral organ. Right, this is basically the sense organ for hearing. So this is how you hear stuff. This is going to be in the middle of the cochlear canal. Then you also have another structure that's important called the basilar membrane, right? So this is basically moves up and down. So basically subject to distortion. And when it distorts and moves up and down, it'll bend the stereocilia of the hair cells, which are embedded in another membrane called the tectorial membrane, which is not subject to distortion. And so when you get that distortion and that movement, you're going to get the activation of the cochlear nerve, which is then going to travel to the auditory complex. This is kind of what it looks like. So here you can see you have the cochlea, that's the snail. When you cut it in cross section, this is what it looks like. So the cochlear canal is this large central region right in here. On the top is what's called the vestibular canal, fluid filled. On the bottom is what's called the tympanic canal, also fluid filled. Here's the cochlear nerve, which basically moves right in underneath these hair cells. So the hair cells in the stereocilia are going to be under here. And so you can see that under here, this is where you're going to see your two membranes, your basilar membrane and your tectorial membrane. And so those are going to be designed to move. So let's go ahead and blow that area up. So this is what it looks like. So this is basically going to be our hair cell. So here's our hair cell here. Notice it's connected to the cochlear nerve. There are other hair cells as well that are also connected to the cochlear nerve. Now the tectorial membrane, which is basically stays in put, versus the basilar membrane. So what's going to happen essentially is with the tectorial membrane is the fluid waves are going to come through the canal, the cochlear canal, and then it's going to distort or change the basilar membrane, which is going to cause these hair cells to bend back and forth. And these are attached to the tectorial membrane, which does not move. So as these guys move laterally back and forth because of the waving basilar membrane underneath them, they're going to basically pull to the side and to the side. Those stereocilia will basically open up the nerve channels, which will then stimulate the cochlear nerve. And so the stronger your wave, the stronger the stimulus. The lighter the wave, the lighter the stimulus. And this is what it looks like. So these are actually the stereocilia in a scanning electron micrograph. So each part of that spiral organ is going to be sensitive to different types of wave frequency or pitch. The tip of the organ responds to low pitches, the base to higher pitches. So basically, the nerve fibers in each region lead to a different area of the context, which will allow you to associate differences in those sounds. So volume then is a function of the amplitude of the sound wave. And the amplitude is the height, right? So if you have something like this, that's a very low amplitude, a soft sound. If you have something like this, that's a very high amplitude, so that's a loud sound. Pitch is associated with your wavelength. For instance, a high pitch is going to be a wavelength that is very tight together. Low pitches are going to be where the peaks and the troughs are very far apart. So it's a long wavelength. So ultimately, what happens is the loud noises will kind of have an effect on the fluid in your head. In particular, in your vestibular canal, which is the upper chamber. Remember, the vestibular canal is the upper chamber. This is going to create more pressure on that basilar membrane. It's going to vibrate to a greater extent. And this is going to be going to your brain for your brain to interpret as volume. Okay. So this is kind of like different types of sounds that you can hear, the sound levels, and the effect. For instance, nightclub thunder, if it's over 120, you're looking at hearing loss. If you're looking at a jet engine or shotgun or rock concert, you're talking about over 125 decibels. This is beyond your pain threshold. Your hearing loss is pretty high. Chainsaws, jackhammers. So this is basically, if you're there for a minute or more, you're at risk. Tractor, newspaper press, subway, motorcycle, basically you have to be exposed here for 15 minutes for harm. A lawnmower or food blender, you're talking about 8 hours for harm. A diesel truck or your average city traffic noise, it's annoying, right? But constant exposure may cause it, but you're talking about really constant exposure, right? Okay, so let's take a look at equilibrium. So equilibrium is basically associated with your vestibular nerve, not your cochlear nerve, and it originates in your semicircular canals, your saccule, and your utricle, which are vestibules that are below the semicircular canal. These guys will take signals to the brainstem and to the cerebellum, and they will interpret that as equilibrium. Other structures that are involved in there are the eyes, for instance, and proprioceptors that will allow you to determine your sense of equilibrium. So let's take a look at rotational equilibrium, your sensation of your ability to rotate. This is going to be handled by the semicircular canals and the mechanoreceptors in the semicircular canals. So this is basically one in each dimension of space. You have semicircular canals in both the X, Y, and Z axis. And at the base of them are little structures called ampulla. These guys contain hair cells as well. Remember, those are those sensory cells we heard about already in the cochlea. They also have stereocilia, but they are embedded within a gelatinous called cupula. We'll take a look at ampulla basically will respond to head rotation in a different plane of space depending on which semicircular canal that it's part of. And so the fluid in the canal itself will flow over the cupula, bending the stereocilia, which will cause the nerve to signal to the brain that you just moved in this direction. And so then ultimately what will happen is you'll put out the appropriate motor output from your skeletal muscles to correct your body's position if say, for instance, you're falling over or something of that nature. So here's what it looks like. So here, essentially at the base of your semicircular canal, or at the bottoms of your semicircular canal, you're going to see your ampulla. So you can see here are your hair cells here. So all these guys right in here are your hair cells. And you can see like the little flame-like stereocilia that are sticking up like little flames. And they're embedded in this gelatinous teardrop, which is the cupula. Now around that is the endolith. This is the fluid of the canal. So as you rotate your head, the fluid of the canal moves to the side because of centripetal force. And then it's going to bend the cupula, which will bend the hair cells, which will then ignite the nerve ending of your vestibular neuron, of your vestibular nerve. So then that's rotational equilibrium. Gravitational is associated with the structures that are just below the semicircular canal, the ventricle and the vacuole. So these are two membranous sacs that are located at the base of the semicircular canal. And so they also contain hair cells. So you can see there's a common pattern here. We also have stereocilia that are also embedded in kind of a gelatinous tube. But in this case, what they have is otoliths in this gelatinous tube, right? So these otoliths are actually calcium carbonate granules. These are rocks. Rocks in your head. That's what these are. These are rocks. Otolith means auto means ear. Lith means rock. These are ear rocks. And so the utricle in particular is sensitive to horizontal movement and bending of the head. The vacuole responds mostly to vertical movement. So you have different axes that should be looking at. So these utricle and vacuole are going to be here. So the utricle is this upper chamber, this upper pouch just below the semicircular canal. The utricle is the smaller pouch just below. Excuse me, the vacuole is the smaller pouch just below. So that's kind of what it looks like. So here you can see you've got your vestibular nerve, which is basically innervating the entire membrane. The hair cells here with their little stereocilia sticking out into this kind of gelatinous mucosal secretion, full of these little rock-like structures, otoliths. And then as basically you tilt gravitationally to one side, gravity will pull all these otoliths to one side, bending. Notice how you're bending your hair cells. Bending your hair cells will activate that, will bend those cilia. That'll activate your vestibular nerve, and you'll basically perceive that as a change in gravitational movement, right? Your gravitational orientation. So here you can see your kinocilium, where your stereocilia is basically bent, and it's going to send that signal to the nerve, and your brain will associate that with a particular movement, right? So basically, as the otoliths move, they're just above the hair cells, but then when you move horizontally or vertically, the otoliths are displaced, and then those membranes will bend the stereocilia and the hair cells underneath, and that will then be activating the nerve, and then that'll send that to the brain. So once the stereocilia bend toward the largest of the stereocilium, which is the kinocilium, this is kind of like a big trigger, then the nerve's impulse will increase, and the vestibular nerve will pick up this nerve impulse and send it off to the brain. And so if the stereocilia move away from the kinocilium, then it's going to reduce that impulse. So ultimately, if you're moving, the frequency of your nerve impulses in the vestibular nerve indicates your movement up or down. So how far it's going up or down is basically how you determine both gravitational equilibrium and your rotational equilibrium. Okay, so that is the end of chapter 15.